There it is. Okay, so we're going to cover the Thalamic Reticular Nucleus. Okay, I'm going to map out some episodes here that kind of hits on this. We talk about it a lot though, because it's going to have to do with the EI imbalance. And this is the protective shell that surrounds the thalamus. We talk about this a lot, so it's going to be nice just to see it all laid out and understand its functions. The main episode that we'll cover is episode 83, so not long ago. Thalamic Reticular Nucleus, sensory gating, and autism. Episode 82 will also hit on this, and that's Sonic Hedgehog. Let's see, and the inhibitory neurons and autism. So we had back-to-back episodes on this inhibition. This is all inhibition. The TRN is all inhibition. And then episode 60 or 76 kind of covered this because this is Parvalbumin interneuron. We'll talk more about this specific interneuron, this inhibitory interneuron, a lot because it makes up the most of this. Parvalbumin interneuron and the autistic phenotype. Episode 84 is the opposite side of this, and that is all about excitation, the go signals, excitatory neurons, the brain's go signal, and autism. Okay, showing up, nah, not bad. Okay, and then way back, episode, let's see, 3 and 4 touched on. It was like a 2-part excitation inhibition episode, back-to-back episodes. Episode 4 would have touched on more inhibition, but it wasn't very specific as these episodes are. These are really dialing in to specific aspects of the excitation and inhibition, especially for this one right here, the Thalamic Reticular Nucleus. Now, science, because people are kind of career-driven, the TRN, you can see it abbreviated and named many different things. Just for the sake of this, just to keep it standard here, keep it similar, we're going to say it's TRN. You can see it probably 3, 4, 5 different names. It's all the same thing though. We love to identify and then name things and then rename things and then rename things. That's just the culture and the kind of the atmosphere of science, because people want to kind of make a buzzword, create this new idea and have it captured, have it gain momentum for funding purposes, for citations, just for the publicity of it. It's irrelevant. So let's talk about the Thalamic Reticular Nucleus. Remember, it's the shell. So let's just draw out the thalamus. Remember, the thalamus is a workhorse. That's why it has kind of these assistants. Remember the basal ganglia episodes, the subthalamic nucleus, which just sits posterior and inferior to the thalamus, and it helps relay the go, no-go signals in the basal ganglia. So let's just draw this out, the thalamus, because we're going to cover, hopefully eventually, the different subdivisions of the thalamus, because there's a lot. The thalamus is a lot to parse out. And there's a good drawing of this, and you can see right here on the daylight. You can get this image on the internet. It's a very good image of it with this shell right here. And then this is helping you parse out the different subdivisions, because remember, this TRN just surrounds the thalamus. And then here's a different view of it. You can see the TRN is all around the outside, prevents signals from coming in. So I'm going to draw that top one out though, just because I think it might help. It might help us understand it. Remember, at the bottom here, you have the medial genicular nucleus right here. Remember the auditory. This is the relay station for audition. Remember the auditory brainstem response, ABR. This is the kind of the last stop. This is where all relay for the auditory happen. And then right next to that, laterally, is the lateral geniculate nucleus. This is the same thing, but for vision. This receives roughly 80%, maybe 75% of the retina's fibers. All vision will eventually come here, but the other 10 to 15% gets the superior colliculi, which is in the mesencephalon, which is just, they're all right here. There's spatial relationships. They can just reach out and high-five each other. which is what I said in the Autism and Eye Movements episode, all about the superior colliculus. So we have the medial and the lateral here. The thalamus is kind of egg-shaped. It's deep in the brain. Remember this area right here. The third ventricle is right here. And then the lateral ventricles are up here. There's a lot of water here. There's a lot of electrical power and things just surrounding this different nuclei surrounding these areas deep in the brainstem, or the brain, just above the brainstem. So the TRN, as you see in that picture, just imagine this is kind of this cut out, and then it's just a shell. It's a shell over it. And then we're going to break this shell down a little bit. Hopefully it helps make sense of it, because there's a lot of cool stuff here. At least I think it's cool. I love this stuff though. So, and like the bottom picture here, okay, you got this on one side, and then you got the thalamus on the other side. Now remember, one side of the brain or the hemisphere is ipsilateral. Both sides is called contralateral. This is just the contralateral. So this is one hemisphere, this is the other hemisphere. And the TRN would be, actually, I think it might look better if we just had some color in it, like Bob Ross. Let's just do it. We're going to get through this, I promise you. See, just basically like this. So, yeah, like the third ventricle will be right here. Before we parse this out, remember the Sonic Hedgehog. Remember we talked about the Sonic Hedgehog a lot. Several different episodes, remember? In neuroscience, in things like rodent studies, you can have these things. So SHH is Sonic Hedgehog. And then you can see in neuroscience papers where you can knock out the gene, protein sequencing, and so forth. So it will look like this. That's a knockout, double knockout. So the animal, the subject, doesn't have this at all. Then we can see what happens. What is missing, or what kind of behavioral effects does this have? It's basically like a lesion, okay? Remember in the Modulation episode, when we are kind of subdividing the neuroepithelial cells, we have the 4 cells in the neuroepithelial, 3 of them for the brain, and we kind of start looking like this, right? We have the telencephalon and the diencephalon here, the mesencephalon, and then the things like the my and metencephalon. I think I have those backwards. Met and myencephalon. Remember, this one does not subdivide, but these are subdividing. So this is the big subcortical area and the cortex right here. And then this is just the little brainstem area. It's a very small area here. And then this is more of the pons and the cerebellum, and then the medulla, and then the spine. If you knock out the Sonic Hedgehog, it's responsible for this area right through here. So in a rodent model with this knocked out Sonic Hedgehog, you don't get this. This does not form. It doesn't really do much for the human autistic phenotype, but at least it provides information on what is causing abnormalities with the Thalamic Reticular Nucleus. Okay, so it's kind of unimportant for this episode, learning about the TRN, this shell, but it's huge. It's a good finding in neuroscience and with the knocking out of that gene and the protein sequencing. So what is the overall function of this, and what makes this? Remember the Parvalbumin interneuron. Okay, we had that specific episode on that, because there was a great paper published about how autistics have kind of problems in a couple of different brain regions with the Parvalbumin interneuron. And remember, Parvalbumin interneuron is the most abundant GABA inhibition neuron that we have. Okay, and this is acting on GABA A, which is very fast acting receptor. It's ionotropic. I mean, these are happening within 1-2 milliseconds. And then the Parvalbumin interneuron makes up the majority of the shell. The second type is Somatostatin. This is the second most abundant GABA cell neuron. And this works on GABA B receptors. These are metabotropic. These are a little bit slower. These are roughly 20 to 50, even longer milliseconds. It takes longer for these to act. Now, what are these two roles in this thalamic reticular nucleus? Is the parvalbumin, which is most of this nuclei. It's acting on specific inhibition. The somatostatin is more like globally. It's just kind of sprinkling the area just to help maintain balance. Remember, we talk a lot about the EI balance. With the autistic phenotype, there's a lack of inhibition. There's problems with inhibition. Not only a reduced number of cells, but the cells on board are kind of abnormal. This is a great finding 2 decades ago, even maybe longer, because something caused Rubinstein and Mike Merzenich to study this. They don't study autism. They study things like neuroplasticity, but something caused them to study this. I don't know what, but it's a good paper. 2003. So what about this TRN? What else besides these two specific cells make up this? We talked about in the episode of the Thalamic Reticular Nucleus about the different sectors. Okay, so the first sector is sensory. The thalamus receives all sensory information coming in, even things that we don't really pay attention to, things that we're not perceiving. That's because of this. So with this, we obviously have visual. So the lateral geniculate is going to be involved. We might as well let's see if this will work out. We'll just try this. Auditory. The orange. It's not bad. Looks like Miami Hurricanes. I don't know about that. I don't know about that now. And then somatosensory. Remember, somatosensory is right here. It's kind of sandwiched in between the sensory motor area and the parietal lobe. The parietal lobe is back here. Kind of talk about the parietal lobe here because it's going to be involved. It's very involved in visual thinking, and this is very involved in visual thinking as well. So now, the somatosensory doesn't have its own little relay station like vision and audition does. These are huge sensory processing kind of mechanisms here, especially vision, right? Remember, a lot of our brain is for vision. And then with the auditory, remember, this is another fascinating thing with the endocochlear potential. Remember, this is the highest DC voltage in the human body, happening in the stria vascularis. I was thinking of stria terminalis, but that's a brain region. So anatomy, man, it's tough. So what are some of the inputs coming in? And then what's the little crosstalk? Because pretty much all regions, we could call this a region, it's the thalamus. All regions will have crosstalk. They will cross and talk within each other. It's very much a thing. So let's talk about the afferents, signals coming in, and the efferents, the signals going out. Remember with the basal ganglia episode though, the thalamus is the main goal. Everything from the basal ganglia is working to modulate, dictate what the thalamus is doing. And that will determine our go, our activation, and our no-go, or inactivation. The whole basal ganglia is for this. And then remember the mini columns. Bless it. Let's just draw out the mini columns again. Remember the mini columns, which are everywhere. Remember layer 4. It's all starlet. Remember the mini columns. Inputs come in from the thalamus to layer 4. And layer 4 will kind of start these mini columns. Remember, this is a mini column, and then this is a mini column. And as you build mini columns, they're macro columns. And then the space in between are neurophils. In the autistic phenotype, they're about half the size of typicals. So they're roughly 40 to 60 microns, which is about, if you shave the thickness of a piece of paper in half. The autistic's neurophil is about that thickness, whereas typicals are closer to the full sheet of paper thickness. Okay? But the thalamus, remember, after it runs through the basal ganglia, and we decide to go and no go because they're simultaneous actions. The thalamus will then speak up to layer 4. remember? And then that will jumpstart and go to 2 and 3 next, because everything about the living organism, as far as like intelligence and memory and ability, experiences, our skill sets and so forth, basically live in layers 2 and 3 of the mini columns. And then it will go down to 5 and 6, both 5 and 6. Remember, 5A and 5B, and then 6A and 6B. 5A is for tonic firing. It's just basically firing based off of the inputs and signals coming in. It kind of just is a baseline activation to keep the living organism present, to keep them active and appropriate, in large part here. 5B is more of that burst firing. Remember, what we're talking about is these burst signals, boom, boom, boom. They fire and then they drop, and then boom, 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 and then they drop, boom, 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 then they drop, right? So we get these bursts right here, and then they're working its way up. This is more activation. And then over time, or you can use like physiological tools, breathing exercises, etc., these will start to drop. And this is in large part, you could think about this on a seesaw, because somewhere through here is tonic. Somewhere through here is baseline, your like resting state. So that's what layer 5 is kind of doing. So how is this involved with the thalamic reticular nucleus? Well, the shell is determining. It's helping regulate this and the actions coming out of the thalamus. What does the thalamus want to pay attention to? What does the living organism want to pay attention to? That way the thalamus can do its work. Remember, it's a workhorse. So let's keep in mind the basal ganglia and these mini columns. I don't have the basal ganglia mapped out here, but the end target is the thalamus. Okay, so the connections coming in, a lot of them are that crosstalk within the thalamus, because the signals will eventually, well, actually, let's talk about, because in the episode 83, I think it was, the one that's specific to the thalamic reticular nucleus, we kind of outlined it as like guards. These are like guards and kind of providing protection. So let's map out what has access and the level of security that this shell is providing the thalamus. What kind of security? What about this information coming in? So just remember, this is a large part sensory processing. Over here. Okay, what, how much, access. Okay, so the sensory, vision, auditory, touch, and so forth. How much access, or said differently, how much security? So I have to make a decision here. Do we want to go how much access or how much security? Let's just say access. Very little. No access. These signals coming in from either vision, auditory, touch. Let's just say the retina is sending some fibers to the lateral geniculate nucleus and then some fibers up to the thalamus. It's going to be regulated by the thalamic reticular nucleus. And then it's also going to send signals back to the visual cortex. So if we want to draw that out real fast, okay, we have the retina. So signals will come in to the lateral geniculate nucleus, roughly 80%, and then about 85%, 15% to the superior colliculus. The lateral geniculate nucleus will send some to inside the thalamus, especially this area right here, which is pulvinar. This is a lot of sensory, but the thalamic reticular nucleus will determine how much and what kind is going to be able to go from the lateral geniculate nucleus to inside the thalamus. And then it will also, the lateral geniculate nucleus will also send fibers, projections, however you want to say it, to the visual cortex, V1 through essentially V5, right? And then it'll just kind of work its way through the occipital lobe. And then the parietal lobe will be up here. So like the brain is kind of like this. And you got the eyeballs right there. The temporal lobe. So you can kind of see this cartoon drawing of retina to the lateral geniculate nucleus here. The lateral genicular nucleus will send some information to the thalamus, most of the information back to the V1. We also have the superior colliculus processing vision. And remember, a large part of this is going back up to the thalamus and to the frontal eye fields. Frontal eye fields are up here. And there's a little subdivision of the thalamus right here, medial dorsal, and that's getting a lot of the information from the superior colliculus. It's a lot, I know, but just to understand this visual information and the processing of it, just kind of follow along here with what this thalamic reticular nucleus is trying to do, because there's a lot of information. Just imagine if you don't have this, or if it's abnormal and you're processing all the sensory information. It's a lot, but you know, it's easy for you to say that the autistic phenotype can be overstimulated. You can understand that easily. This is what's going on. That's why I want to cover this stuff. That and I love it. Okay, so the sensory. It has little access into the thalamus. This layer right here, remember, 6A and 6B, one is cortical, bless it. Remember the white fibers right through here too, like the fasciculi and so forth. So cortical, cortex, or cortical. So this is just speaking back and forth through the cortex, like the parietal lobe will be heavily involved, the sensory motor and the prefrontal. That's what this is. After layers come down from 2 and 3, sending signals down to these cells, and then the white matter tracks here, there's a lot of crosstalk between the cortex, bringing other subdivisions on board, helping the living organism adapt responsibly. And then the other one is cortico Thalamus or thalamic. This is a big one here, okay? Because the mini columns are already receiving information from the thalamus into layer 4 with these stellate cells, and then it goes up to layer 2 and 3 to gain information about what the living organism knows. Everything the living organism knows is basically receiving a summary here, okay? This is what's happening from the environment as far as sensory processing. What do I do? And remember, in the background, the basal ganglia is working to help determine movements, because the basal ganglia is movements, and that's the living organism. That's the role of the central nervous system, is to move the living organism. We're mapping out how it happens. So the thalamus is sending cells or information to layer 4, and then it goes up to layers 2 and 3 to receive information, and then that information is carried down to layers 5 and 6. And from 5 and 6, it's kind of being a completion, but it's never complete. It's just updating, because it's a constant loop, man. It's just constant. And what's allowing the thalamus to be updated here is this layer, corticothalamic or thalamic, because based off of everything coming in, it's getting updated. How is it doing? What does it need to do? How is it making adjustments? And that's in large part what this is doing too. So let's just imagine this is the parietal lobe, where sensory integration is kind of happening. Higher order sensory integration, making meaning, making sense of the things in the environment. And then it can send signals to either the sensory motor, because maybe you just have a habit based off of this information. That habit will then send information down to the dorsal striatum, remember the putamen. Remember, we mapped out, we extracted a specific cell from the putamen, because this is where habits happen. Sensory motor to the putamen, that's very lateral in the dorsal striatum, and a specific cell spine will hit. Remember, these are medium spiny neurons. We've mapped this out, and then it will orchestrate based off of the basal ganglia, those other relay stations and output areas, and it will inform the thalamus. Okay, what am I doing? Remember the habits, though. You don't have to think about things. You just automate responses. That's why we love habits. If it's a habit, this is just an example of a habit. Let's say it's not. So this cortical could send information to the prefrontal cortex. Maybe it's parts of the dorsal lateral, which is this superior colliculi will update the dorsal lateral early. Well, let's just map it out, because I really want to talk about the prefrontal cortex now, for sure. So we have the eye again, okay? And then the thalamus here. And then we'll do the brainstem here. You know, remember we have the pons, the little football shape. Then we have the cerebellum, superior colliculi, inferior colliculi, right? And then we have the occipital lobe, the parietal lobe, sensory motor, we're up through here now. The dorsolateral, we're getting into the dorsolateral area. This is going to be more of the orbital frontal, and just to help us make sense of where we are, okay? Remember, the basal ganglia is right through here. Remember, it's all surrounding this ventricular area, the third and the lateral ventriculars, okay? A lot of electrical signals here because there's a lot of water. There's a lot of freaking water there. Low blood-brain barrier, too. So let's just map out this prefrontal cortex. Let's go blue right here. Dorsolateral. Use these colors if I don't have to. I don't know if that shows up much different than black. It kind of does. Orbital frontal cortex. This is why I want to parse this out, because this is a lot of sensory. It gets fast-acting things from the environment. About 90% of the things in the orbital frontal cortex, which is just right here, just right on the other side of your eyebrows, is sensory. It's very specific as well. And then deep inside, on the inside of this area, is the medial prefrontal cortex. We've talked a lot about that, especially the anterior cingulate cortex, okay? We'll do an episode all about this because these are heavily involved, especially the medial prefrontal cortex, and maybe more so the orbital frontal cortex and the dorsolateral prefrontal cortex more than what people might consider. I know the medial prefrontal cortex is heavily involved with autism because there's a lot of studies done, because this is where adaptive responses happen. This is where, as we're navigating the world and we're mapping this out neurally with these connections, the medial prefrontal cortex leads the way. And in large part, it's these other subdivisions of the prefrontal cortex are helping out. The anterior cingulate cortex. Remember, they have the spindle neurons and the lack of Parvalbumin. So the medial prefrontal cortex there's a lot of different abnormal cells, neurons here. That's why it's really known. And in addition, the adaptive responses, because everybody knows that the autistic in a social situation lacks kind of skills, because this is being suppressed, and these other subcortical areas down through here, and the habit formation with the rigid thinking, the repetitive behaviors are acting more than here. This is more habit. This is more goal-directed. This is more being able to adapt in the environment based off of useful skills. So that's big. Okay, let's just get back to what has access now. I'm trying. We're getting through it though, because we were talking about the layer 6. So this information coming back down here, back down to the thalamus, is not very good access. Little access. Okay, so we just mapped all this out, receiving sensory information from the environment, which is that's what sensory processing is doing. It's extracting physical information and phenomena from the environment, so the living organism knows what to do. We mapped all that out, and then we sent it up to the mini columns, whichever cortex or whichever area you want to think about. They're all slightly different. In large part, what's different is the layer 4, and then what kind of cells make up layers 5 and 6 and 2 and 3. A lot of pyramidal neurons and then inhibition neurons are making up layers 2 and 3 and 5 and 6. And then you have things like microglia, like the little support cells, which we haven't spent a whole lot of time on that, I admit. So layer 6 has very little access. Layer 5 has a lot of access. Remember, this is more firing rates. Good access. Lots of access. Not bad. Okay. This tonic and burst firing will help determine, because this is kind of providing some modulatory impact, some intensity. How much intensity? So that's valuable information. And then we have some access from the brainstem, especially remember the auditory brainstem response. But there's other things. Remember with the right here with the brainstem, there's a network called the reticular formation, not reticular TRN. It's just a repurposed word, reticular. reticular activating system is one, where it has ascending and descending fibers. And just the reticular formation, or just a collection of these brainstem areas used for arousal, used to go activate the sympathetic nervous system. So this is kind of involved with that. So it has some brainstem arousal. Get up and go. The axis here with this reticular activating system is context dependent. So it's hard to say if it has easy access, you can just get right in, no security, or if it's heavily guarded. It's context dependent. Okay. And then there are things like with this crosstalk. How much does this shell have to be involved with this crosstalk? Maybe it's talking back and forth to these different subdivisions here. There's a lot of different subdivisions in the thalamus. It's a heavily studied area here. So this kind of helps with what has access. The big thing to remember here is just the raw sensory input here has very little access. Now, one thing that I should mention is things like pain, things coming up from the body, that intensity is also more context, but mostly pain has pretty strong access. So there's not a lot of regulation control by the TRN. And here with this pulvinar, which is heavily involved in sensory information here, but the difference between the sensory sector of the shell and the subdivision with the sensory processing inside the thalamus is this provides more attention. So remember the salience network. Remember the superior colliculi reaching up to the frontal eye fields and then back down to the thalamus and back down to the superior colliculi. This information here regarding the sensation isn't so much raw anymore. The living organism is making sense of it. So that visual input has little impact for the Thalamic Reticular Nucleus. It has direct access to go back into the thalamus. Why? Because it's determined. Something's made the determination that that's valuable, because that's what I'm spending my spotlight of attention to. Remember, we've talked about signal to noise, and this is what I want to talk about kind of right now because of how important this is with the signal to noise, because that's a lot about what we're talking about. So I'm just going to go ahead and erase some of this stuff here. So we can talk a little bit about some of the functions and goals here. Sensory gating, it's already covered. We already did a pretty good job of covering that. Okay. And then the brainwave generation. Remember the layer 5. The layer 5 helps with this. And something that's pretty good is alpha band. MIT does good work on alpha, or the brain waves, and it should probably have a little bit more traction, I think, in research is the brain waves. But I don't know. The brain wave is very functional, especially alpha in this regard, which is 8 to 13 hertz. Remember, we kind of covered that. You could probably pick up, based off of what we've talked about, attention modulation. This is helping. So it's unclear to me if this process is helping attention modulation, or if I think it's probably bidirectional, especially if you think about the autistic phenotype. It seems like it's bidirectional. Things that are irrelevant are very much irrelevant to the autistic phenotype. Things that are salient are very much salient to the autistic phenotype. So I think this, and with that lack of adaptive responses and so forth, and the habits and the restricted fixated interests that are abnormal in intensity or focus, this is kind of helping explain why the autistic phenotype has that. What is really a big wow factor here for the autistic phenotype and this so-called sensory gate is regulating sleep-wake states. This is not talked about enough in the autistic phenotype, but every autistic phenotype, every person with autism has sleep-wake dysregulation. These things, 100% of the autistic phenotypes have sensory processing problems, which we're covering. In large part, this is the core of it. 2 is speech language problems. 3, GI problems. 4th, and an underrated one, circadian rhythms. E or no E? I don't know. Could be an E. But these 4, 100% of autistics have. 100%. 100%. I don't care what the research suggests. Like this 50 to 80% of autistics have a sleep-wake disorder. No, it's not. It's 100. I promise you. And GI and speech and language and sensory processing. I promise. And a 5th thing would be mitochondria. But semantics. I'm not going there. The sleep-wake regulation, remember, in large part because of this area right here, the reticular activating system is involved with that. This brainstem here is a superhighway, man. And it's got a lot to do with balancing the brain and physiology. Okay, so sleep-wake, not bad. And another one is just overload protection. It's just that protection. Like, especially said the same as, I think, maybe 1 and 3. But it's a big deal. It's a very big deal. Another thing that I want to talk about while we're talking about this is back to the rodent knockout model, is because this right here is heavily involved in autism, CNTNAP2. You can double knock out this, and you're going to have hyperactivity because of this is going to be abnormal. This is very well studied in autism. I don't mind this as like one of them so-called gene things. I think genes are broken. But, there are some, like this one, and Shank3, neuroligin, nororexin are good ones. P10, good one. It's more of an enzyme, whatever, but some good stuff there. So whenever this is knocked out, you get the hyperactivity, you get seizures, and poor socialness. Poor socialness. Wonder why? Or this is underregulated. There's a lot of information coming in and through this. Makes it hard for the environment. Makes it hard to interact. Remember listening dissonance. Remember the processes with the auditory brain stem response with wave 5 and the vision aspects of it. There's a lot happening here that's abnormal for the autistic phenotype. So there's isolation in a rodent knockout of this right here, and also repetitive behaviors. So what does repetitive behaviors look like for a mouse or a rat? A lot of licking, doing just a lot of the same kind of behavioral activation, the same action, just abnormal than what they were doing or what their peers are doing. So this is a pretty good finding too. What's different about this and the Sonic Hedgehog, I think, is because this comes on early with like the neurulation. This is more downstream. This is more after. So it's not likely that the human living organism is going to just not have the shell. That's not likely to happen. That's not likely the case. But it is likely, because we know that, remember from the Parvalbumin even, that we can have a shell, but it's just not functioning properly. So this is more in line with that. And based off of these kind of phenotypes from this double knockout, it's pretty good data. So what happens? Let's just end on what are some of the strengths of this kind of abnormal shell. So if you think about it, if you think about what we've been talking about, there's a lot of information coming in. So what is a lot of information? What could that mean? Well, we know that accelerated learning, okay? So strengths of a poor TRN, it's accelerated learning. Why? Because of that attention modulation is inner. We're inward. The autism, look here. Autism comes from the word autos. Autos, like automatic, automobile. This means self. Too few people know this. More people need to know autism means self. So accelerated learning, because if you think about learning, you need time on task. You need access to the information. This actually provides more. So this is a huge thing. We talk about these splinter skills and so forth. I don't necessarily know if I like the term of that, but with having a poor thalamic reticular nucleus, we can accelerate the exposure. And this is good for learning. And it's not difficult to see, especially if you read the early Tanner papers from 1943 or Asperger's paper from 1944. Asperger spent a whole lot of time, those are long papers, by the way. Asperger spent a lot of time on autistic intelligence, and Kenner did too. He just didn't frame it with those words. Asperger called those little kids little professors. This was happening. This was happening back then. So this accelerated learning is a very functional thing. The second thing, I don't want to say too much about this right now because it's at least one episode. This is very hard to explain if it's not you. But that is visual thinking. It's almost like this is bidirectional here too. And then you can lump things like this on. But remember, right here is the parietal lobe, and then there's a little nuclei here called the precuneus. The precuneus is involved in the default mode network as well. Where am I? Right here. The default mode network, okay? And visual thinking. This is imagination. When sensory information, the sensory processing it's heightened in the autistic phenotype, remember, because there's a lot of brain waves being responsible here, and even independent from the shell, independent of it, with the retina to the lateral geniculate and the superior colliculus, and the lateral geniculate and the superior colliculus both are sending fibers to the visual cortex, right? Because the retina, it's split between sending some projections to the lateral geniculate and the superior colliculus. So there's like two distinct pathways here, but each of these two will send some information up to the visual cortex. We'll parse this out. The visual cortex is being bombarded, and it makes it hard to kind of flow through the process. And that's why you hear of things like bottom up or details first. This is so huge with learning. This is so underrated, and this is a big benefit. This is just such a huge benefit. I can't speak enough on visual thinking and accelerated learning, or even the default mode network, because that's a big misnomer. Science and people think the autistic phenotype has an abnormal default mode network. No, this is our default. The default is the default mode network because it's all in-ear or inner. Remember the salience network as well. Inner. Autism means autos, which means self. It's not complicated stuff. People researching autism are not autistic. People researching autism are just viewing it from the outside. The weaknesses, though, we kind of talked about this. The weaknesses is the hyperactivity and just the lack of being able to filter out properly. So there's like anything in life. There's some benefits and some struggles with the thalamic reticular nucleus. But if you have the thalamus like this, the shell over it for protection, thalamic reticular nucleus.